So I'd like to welcome you to the 13th International Union of Immunological Societies and Frontiers webinar on COVID-19. My name is Nigel Stevenson, and I lead the Viral Immunology research group at Trinity College in Dublin. And today it's my great pleasure to welcome our 13th speaker, Professor Adrian Hayday from King's College London. You're very welcome, Adrian.
Thank you, Nigel.
So Professor Hayday started studying immunology at MIT, where he and his colleagues first described the wholly unanticipated at the time gamma chain of the T-cell receptor. Professor Hayday has authored over 200 papers. He's been elected as the leader of the British Society of Immunology between 2005 and 2009. He's a fellow of the Academy of Medical Sciences and the Royal Society. And currently, Professor Hayday is Professor of Immunobiology in King's College London. Professor Hayday is also a clinical academic group leader at King's Health Partners and is a group leader at the Francis Crick Institute, where he's Assistant Research Director. So today, before I invite Professor Hayday to speak, I'd like to encourage you all to submit questions throughout the webinar, which I will put to Professor Hayday in a discussion question and answer session after his presentation. So without a further ado, I'd like to invite Professor Hayday to tell us about his exciting COVID-19 research and his phenotype project that he's been carrying out in King's College London.
Thank you, Nigel. I think we will be expecting the slides any moment. Yep.
There you go.
Great. Thank you very much.
Over to you, Adrian.
Yes, well, I'd like to thank everybody for the opportunity to speak to you. When this was scheduled some weeks back, I was rather hoping that we might be through some of the worst experiences of COVID-19, but the data that are coming through from the WHO globally tell us that, in fact, we remain in the midst of an extremely serious global emergency. And so it's a pleasure to talk to you, in fact, an honor to talk to you about a very small contribution that we've been making to what has been an extraordinary global and collegial effort. We call this COVID-IP. It's an immunophenotyping project, and we are seeking to understand correlates of protection and pathology in COVID-19 disease. The project has only been possible because of two key co-investigators. Jonathan Edgeworth runs the Center for Infectious Disease Research at Guy's and St Thomas's Hospital in King's College, and Manu Shankar is an immunologist and also a consultant in intensive care medicine. So COVID-IP has sampled 128 subjects. And just to make this clear from the beginning, there are 63 hospital-treated COVID-19 patients who have been examined during their disease course, and they have been segregated according to the WHO ordinal scale into 6 low-severity patients, 26 moderate severity, and 31 with severe disease. They've been compared with 55 controls, of whom 23 are actually seropositive because of prior exposure to SARS-CoV-2. 8 of those were symptomatic but did not require hospitalization. 15 were asymptomatic. And another set of controls are 10 contemporaneously hospitalized SARS-CoV-2 negative lower respiratory tract infection patients of a similar age range and comorbidity spectrum as the COVID-19 patients. And we have looked and a large number of parameters, cell numbers, cell frequencies, cell phenotypes, exhaustion, activation, cycling in the peripheral blood. We've looked at serum analytes, antibodies, virus burdens, and we're in the process of looking at antigen receptor sequencing and antigen-specific functional responsiveness. And as you can imagine, I will not make any attempt to go through all of those things today. But what I'd like to go through are a few key points, and I'd like to start by considering the rationale for the study. So, of course, SARS-CoV-2 is essentially a new pathogen for human beings, and as is true in all infectious disease, the key is to understand the host-pathogen relationship, at the core of which there is the immune response. If we can understand the immune response that is ongoing during COVID-19 disease, we may be able to better understand the disease and better manage the disease, and thereby reduce the global threat of SARS-CoV-2, whether or not we have an easily accessible vaccine. And third, this is a very rare opportunity to study an ongoing immune response during human disease. Now, there are counter considerations. This is, of course, not a study of the most efficacious immune responses to SARS-CoV-2, because we are not accessing and studying asymptomatic individuals who are infected and shed the virus. It's also true that in our patients, neither the timing nor the dose, neither the repetitiveness nor the routes of infection are actually known to us. And there is, of course, enormous patient heterogeneity. St. Thomas's Hospital is an area of very mixed socioeconomic and ethnicity, and there are all sorts of ways in which the patient cohort is heterogeneous. Nonetheless, we have a hypothesis, and the hypothesis is that despite all of that, there may be an overarching immune response signature that will emerge above the noise of heterogeneity, just as many papers over the last decade showed that enormously diverse people are capable of making very similar immune response signatures when, for example, challenged by vaccination. So, to pursue that hypothesis, we adopted a high throughput approach. This, for example, is illustrated in the paper that we published just at the beginning of the year, which was a high throughput approach to mouse immune genetics. And in that sort of approach, the whole goal is to learn as much as you need to make certain conclusions, but not to aim to learn more at this stage. All of the effort and all of the efficiencies should be invested in the robust performance of standard operating procedures. We have, through that high throughput analysis, collected about 600 data points per sample, giving us a total now of over 100,000 data points. And those data points were derived from widely applicable standard operating protocols, primarily 8 flow cytometry panels that we believe could be rolled out globally and thereby improve the chances for straightforward meta-analysis as the international community seeks to get a hold on what is going on in this disease. Blood analyses don't sample tissues particularly well, but they're highly suitable for longitudinal studies. And of course, information on what's going on in the tissues is reflected in the activation state of peripheral blood cells. We applied very stringent quality controls and stringent statistical analyses, and we aim to rapidly disseminate the data as quickly as possible to the community. And in that regard, we have opened this website where all of the data are deposited and where you can learn about all of our methods. That's www.immunophenotype.org. And I encourage each and everyone to access that to their own convenience. So the cohort of 63 patients is, as I mentioned, highly heterogeneous. If we look, for example, at some important metrics here, this is BMI. Here's a cutoff of 30, which is widely regarded as obese. But you see actually here 3 individuals who have very severe disease, 2 of whom are Black and minority ethnic, despite the fact that their BMI is moderately favorable. By contrast, you have 2 very mild disease cases here with at-risk BMI. And over here, you have, for example, mild disease despite the co-presentation of chronic obstructive pulmonary disease and hypertension, again, in 2 BMAE individuals. So these kind of underlying features, these comorbidities, really don't segregate the patients. But by contrast, what does segregate the patients is an immune signature, as judged by this principal component analysis, purely focused on immune cell phenotypes. So we're not looking here at antibodies or cytokines, we're merely looking at cellular phenotypes, and you can see that the COVID-19 patients, irrespective of their severity classification, have essentially segregated right away from the controls. By contrast, the controls that include both seropositive and seronegative in dark blue, seropositive in light blue, cluster together. So at this point in their sampling, their cellular immune responses, as judged in the blood, are comparable. So what underpins this COVID immune signature? So to look at that, we then undertook an effect size analysis, but now including antibodies and serum analytes. And you can see right away that key to the segregation of these individuals from the control group are not only the antibodies, which one will predict to segregate them, but in addition, some key chemokines, IP10, CXCL10, IL-6, IL-8, and IL-10. There's also the clear presence of activated T cells and cycling T cells in the blood. We'll talk more about that in a moment. And on the other side, there is a loss of certain key populations, in particular T cells, including naive CD8 T cells and the prime subset of blood gamma delta T cells known as V gamma 9, V delta 2 T cells. So there is a signature. The question is, how does that signature potentially relate to disease? And so it's now time to look more clearly at issues that may relate to disease severity. So the first thing is to establish that virus burden, at least as judged by clinical sample and controlled for sample adequacy, declines are given days from symptom onset, which would be predicted, but it really doesn't correlate at all either with peak WHO score severity or admission severity. And it really doesn't correlate very well at all with almost any immunological marker, except, interestingly enough, for 2 activated V delta 1 T cells, which have previously been implicated in antiviral responses, and NK cells likewise, as represented in the PBMC compartment. So one of the things that is almost universal across all of the patients is the production of antibodies, which have been assayed both by an ELISA assay and a luciferase-linked immunoprecipitation assay. Our 2 collaborators there, Katie Dawes in infectious diseases at King's College London, and Pert Pettersson at the University of Tartu in Estonia. Now, these are ELISA data, and this is, for example, IgG against the receptor binding domain, and you see very high levels of antibodies in moderate and severely affected patients. But actually, you can see some of these also in seropositive individuals who did not require hospitalization. And actually, the correlation is more likely to be with the length of time available to sample for the development of assays rather than the status of the patients. So in fact, you see down here 3 individuals who don't seem to have antibodies, but they were really sampled very early. And if we now look at time of onset for these patients, we find, of course, that the antibodies accumulate over time, which is not at all surprising, looking at the end of 1 week through 2 and 3 weeks post the onset of symptoms, whereas the frequency of plasmablast expansion that is likely to give rise to these antibodies occurs early, peaks, and begins to disappear. The fact that we could capture this in all of our COVID-19 patients tells us that their signature is a dynamic one. It's an ongoing B-cell response during the disease process. There is some correlation of plasmablast numbers here with severity, but the core B-cell phenotype essentially applies across all COVID-19 patients. Now, if we go back to vaccination studies where people have seen, for example, these plasmablast extensions followed by immunoglobulin production, they were characteristically preceded on day 1 by a significant increase in IP10. This is in healthy individuals. And indeed, if we look at IP10, it's strikingly elevated in COVID-19 patients, but not, for example, in a non-COVID lower respiratory tract infectious patients hospitalized at the same time. Now, this is a striking increase, and as you can see, it's also severity related. And this is an interesting fact because elevated IP10 is actually common to SARS-1 and to MERS. Some of you may know the MERS virus actually interacts directly with an ectopeptidase which regulates IP10 levels. But what is interesting is that although IP10 correlates with interferon gamma, one of the molecules that can induce IP10, its levels are sustained, whereas the levels of interferon gamma and particularly interferon alpha, another inducer of IP10, are extremely transient. So again, you see this very dynamic response in the COVID patients, plenty of interferon alpha here, but a very sustained IP10 production. Now, it's been said that these patients suffer from cytokine storms. In our own experience, we see very little evidence for that. We see very little evidence of sustained production of, as it were, inflammasome-associated cytokines such as TNF or interleukin-1. But we do see very significantly elevated levels of IL-8 that are, again, also seen, for example, in the lower respiratory tract infection controls. We see to some extent IL-6. There's a lot of patients who remain in the normal range, but some are very high. Unlike IL-8, this again seems to be severity related, just as the IP10 was. You can see the IL-8 really is pretty comparable, irrespective of the status of the disease severity. And also related to severity is elevated levels of IL-10. So actually what forms in COVID-19 is what we call a cytokine-chemokine triad, where you see increased levels of IL-10 and IL-6, and in this direction, IP-10, all correlating in a way that you just don't see in a lot of other respiratory tract infections of that kind. So does this have an impact on the cellular milieu? Well, IL-8 is a major factor for neutrophils, and there is some increase for sure in neutrophils. Eosinophils in our hands remain rather stable. But what are really striking are extraordinary depletions that are very severity related of plasmacytoid with dendritic cells and basophils. And lo and behold, the basophil depletion correlates almost perfectly with the increased levels of IP-10. Now, this is fascinating for a number of reasons. Basophils have been implicated in the past in the regulation of coagulation. Coagulopathies, as you may know, are a major indicator of COVID-19 disease. And these basophils are also known to track to the lung, and their depletion from the blood may indeed reflect homing to the tissues. Now, another change in peripheral blood cell numbers that has been widely reported in this disease is the overall change in lymphocytes. In our hands, this is almost exclusively a T cytopenia, and it's highly subset specific. So, for example, here, if you look again at the controls here, these are the seronegative and the seropositive controls, you see significantly reduced numbers of Th1 cells. These are counts per mil, and particularly Th17.1 cells. Again, this is not unique to COVID-19. But the Th2 cells are largely unaffected, and the Treg cells are also pretty unaffected. This is strikingly reminiscent of work undertaken by my collaborator Manu Shankar, who, as I mentioned, is an intensive care specialist. And in his sepsis work, he sees this striking reduction in the representation of Th1 cells and a compensating increase in the representation of Th2 cells because their numbers are barely touched. And again, Treg cells remain fairly comparable. To my knowledge, the reasons underpinning this subset specificity are really quite unknown. CD8 T cell numbers get hit particularly hard, and that includes the naive T cells here. So it's hard to attribute this all to activation-induced cell death or to homing. It is, of course, possible these cells are differentiating into later stages, but their numbers certainly are very severely affected. But this is also true for central memory T cells and also for effector memory T cells. As I mentioned earlier, gamma delta T cells are also very severely depleted. We don't believe that this is obviously explained by homing, not only for the reasons I mentioned, the impact on naive T cells, but if you look, for example, in SARS-CoV-2 histopathology, the infiltration of T cells is not always particularly apparent. And in autopsies, in fact, the most striking phenotype appears to be necrosis in the T cell zones in the spleen and the lymph nodes, not unlike what has been seen in autopsies of AIDS patients. So what's going on with these T cells? Well, a recently developed methodology by a collaborator of mine, Francesca De Rosa from the CNR in Italy, has shown that we can measure cell cycling in the blood of cells that would ordinarily be quiescent cells in transit. Healthy individuals, seronegative or seropositive, have very few T cells in SG2M, whereas you can see here in CD4 and CD8 effector memory cells, very significant numbers of cycling cells that are increasing with severity, just as their activation status is as measured by CD38 and HLA-DR. And if you look on nanostring, these cells show high expression of caspase-3, for example, relative to effector memory cells that you can find as circulating in seropositive or seronegative control patients. And this is also coincident with the appearance of cells which are strikingly double positive for PD-1 and TIM3, commonly regarded as exhaustion markers. So there is this very overt coexistence of T cytopenia, activation, cycling, and exhaustion. Is this true of antigen-specific cells or bystander cells as well? Simply to say that we've now got peptide stimulation assays going where we're able to find these are seropositive individuals who had not needed to be hospitalized, probably experiencing infection about 3 months ago. We still see today residual peptide responsive T cells that we will in future now be able to analyze for their status in asymptomatic or post-symptomatic individuals versus those experiencing disease and requiring hospitalization. So my conclusions and correlates are that across a highly heterogeneous COVID-19 cohort, an immune signature was clearly discernible. It's a dynamic signature with very specific traits, but it's not chaotic. There is an interesting correlate of V delta 1 and NK cells with the virus that might reflect direct interactions with virus-infected cells. But most strikingly, the immune signature seems to blend traits that you see in other settings in a sort of unique combination. The shared traits can guide treatment. So, for example, the subset-specific T-cell depletions that we see in sepsis might suggest the use of IL-7 to restore T-cell function, or anti-IL-6, for example, that is shared with autoinflammatory diseases could be used to restore protection and reduce immunopathology. The COVID-19 severity-related traits may offer insight into pathogenesis. I've mentioned basophils in coagulation, and possibly IP10 levels are common to severely pathogenic coronavirus infections. But finally, I just want to add that some such traits may aid early risk-based stratification, which we believe to be a tremendously important issue. So the elephant, as it were, in the hospital is the fact that this whole disease is taking place against the background of extremely pressured resources. So we looked at individuals and measured their admission parameters for example, C-reactive protein versus IP10. And this graph segregates those according to patients who improved thereafter, 7 days later, or did not improve or even got worse. And what you can see is actually your first bleed IP10 levels pretty much segregate out people who are going to improve. And actually this triad of IL-6, IL-10, and IP10 is an extremely powerful anticipator of that. And we can take this further and look at a key component of healthcare provision, which is hospital stay. CRP is really not a good predictor of how long you're going to stay in hospital. Remarkably enough, first bleed IP10 actually is. So we hope that there may be some capacity to use these immune signatures to aid early risk-based stratification and particularly be better prepared either for a second wave or a similar sort of pathology that may yet emerge from another virus. There are very, very many people to thank. I really can't thank them all, but the good news is all of their names are on the website, which I again draw your attention to, as are the names of the founders and funders. And I'd just like to thank, finally, the patients and particularly the research nurses who went to great risks during periods of immense discomfort to facilitate this study. Finally, I'd like to thank you for listening and thank Nigel for chairing the meeting.
Thanks, Adrian. That was brilliant, really insightful, and such precious resources and patience as well. Unfortunately, my internet died a minute ago, so I was out of the room, panicking that I wouldn't get back. But we have lots of questions here. And the first question, actually, it's really interesting. And as you know, we work on interferon and are fascinated with that. And does the virus load actually correlate to the interferon levels?
Yeah, so it's interestingly enough, it's a hard question to answer because, as you may know, interferon itself, type 1 interferons, are very difficult to measure. But there is, we've been applying a newly developed, highly sensitive technique that was really taken forward by Dara Duffy and Yannick Crow and his colleagues, using antibodies that can pick up type 1 interferons at highly sensitive levels. So by doing that, it's become clear to us that most of these patients do indeed make decent levels of interferon, but it's extremely transient. So the question is, Nigel, is to try to match up the synchrony of the viral load with the interferon peaks, and that's ongoing. But it's an important and a good question. Yeah.
That's interesting. And then there's another one. Just, did you look at anything in relation to type 3 interferons?
No, I knew that would come up, and I was hoping to preempt the question, but I had too many other things to think about. No, we have not done that yet to any degree of satisfaction, but type 3 interferon deserves a lot of attention.
Yeah, it does, definitely. And in relation to the IL-10 that you saw, so there's really high levels of IL-10, but in reality, it's not working. Is that it?
Well, of course, IL-10, like all of these cytokines, has many facets to it. So, of course, it's thought of as an immunosuppressive cytokine. But it's also, of course, it has effector functions. I mean, obviously, it's capable, for example, of promoting B-cell class switching to IgA, which may be extremely important in this disease. I think one of the things that's been unusual for us in this disease, in studying human beings, is that we get the opportunity to see that these levels are sustained. And I think that raises an important question. One can think about the functions of cytokines and chemokines as they're seen in physiology, in which settings they're normally transient with multiple feedback loops and a whole series of events that peak and then decline. I think it's a very different question to ask, what is the function of these molecules when they're sustained at very high levels? And I think, there's all sorts of scope for example, simply for animal models where the levels, but also the time periods, the durability at which cytokines and chemokines are overexpressed in combination. So, for example, overexpressing CXCL10, overexpressing IL-6 and IL-10. What would happen in a mouse if you just maintain those three together for long periods of time? Do you get something that looks like COVID-19 disease, but without a virus? So I think these are fundamental questions that need answering.
That is a fascinating idea. I suppose that links to another question here we have about, in reality, your samples are from blood. So do we have, or do you have any link or any knowledge now of how this might be cells, they're circulating in the blood, but in relation to their location in the lungs, but also in the intestine?
Yeah. So we haven't looked in the intestine at all. What I can tell you, so having done a little bit of skin immunology in my past life, if you look in the blood, you can detect skin homing T cells pretty easily because they're all CLA positive. And likewise, if you look for cells which are going to home, for example, to the blood, they'll be CCR9 positive, et cetera, et cetera. So what we have done is we've compared the T cells in the COVID-19 cohort to the healthy control cohort for differential expression of chemokine receptors. We actually asked it in an unbiased fashion and asked whether any of the chemokine receptors would segregate those groups, and they do not. So if there is a lot of homing to the tissues, I don't think it's the overwhelming feature that explains this complete change in the physiology of the blood T cell compartment. And likewise, I revisit again these observations that are being made at post-mortem of necrotic regions of the spleen and the pulmonary hilar lymph nodes that really are quite reminiscent of AIDS pathology and T cell death. So clearly, this is not to rule out homing in any way, shape, or form. There's certainly a large bialar monocytic infiltrate into the lung, but we think that over and above that, there is a major T cell pathology going on here, which may, of course, be severely affecting the capacity for T cells to be protective.
Adrian, you mentioned this looking reminiscent of some other viruses.
Yeah.
In reality, have we ever seen anything like this immune response, or is really the unique thing its global infectivity and its spread in today's travel, and maybe this being a unique virus jumping from animals to humans?
Well, I mean, it's very hard to answer, Nigel. Of course, it's a question that we all struggle with. But I mean, to me, this may be simplistic, but I think if I go to a clinical conference, what's clear about COVID-19 disease is it rather uniquely mixes lots and lots of things we've seen individually before. So it mixes acute respiratory distress with vasculopathy, which it mixes with renal failure, which it mixes with coagulation deficits. And likewise, I think immunologically, what it's doing is it's mixing a lot of things we've seen before. The B cell response, to me, looks just like one that you'd want if we vaccinated against seasonal flu. And by the way, I would recommend everybody gets vaccinated against seasonal flu as quickly as possible. But then again, the T cell response, which was highly subset specific, looked very much like sepsis. And the IL-6 status and IL-10 status looks like sepsis. So there are all sorts of parallels, obviously, with chronic severe flu infection when it becomes requiring for hospital treatment. You yourself have mentioned before HCV. So what I think here is there's an extraordinary blend. And why that blend is occurring, what are the major drivers of that, and where are the key intersection points we can be effective in managing this disease, I think are the key questions. That's why I think understanding this immunological signature can genuinely be of benefit.
Yeah, that's why I think a study like what you have done with so many patients is so beautiful and so useful.
Thank you.
Really point us towards what to look for. And of course, in China, we'd seen a lot of these, the increase in neutrophils, the decrease in B cells, etc. But it's all this information coming together, so precious.
Yeah, and I think, Nigel, it's interesting because what we've been hammering away at is what we like to call a core signature. So we do see increases in neutrophils. I have to say, we don't see the massive increases in neutrophils that some other people have seen. We don't see changes in eosinophils that other people have seen. So I think there will be, according to the setting and potential for co-infection, there will be variation across the spectrum.
Yeah.
But the key, I think, to both pathogenesis and potentially for early patient stratification is to try to get at these key parameters that just seem to be showing up over and over and over again, despite, and I return to this extraordinary heterogeneity in the patient cohort. So, yeah.
Yeah. And do you think, and in your group, in relation to the neutrophils, were there secondary infections? So some people have postulated that a lot of the neutrophil stuff is because people maybe weren't treated quickly enough in the early days, and they then developed a bacterial infection as well.
So I think one of the reasons that our neutrophil spectrum may be on the low side is because actually that was controlled for quite well in our cohort, and essentially all of the bleeds were done, particularly in the ICU, before any detectable bacterial co-infections set in. Having said all of that, of course, who knows what the hell some commensal that's being dysregulated is doing, something we would never be measuring. So, one just cannot rule things out in this extraordinary complex experimental setting.
Yeah. Just taking back to a question here, they're asking about T cell exhaustion.
Yeah.
Is that something you've thought a bit about?
Yeah. So exhaustion markers come up, and as I mention, I realize this was very fleeting, but again, I can encourage you to go to the website. You'll find everything there. There's a lot of PD-1 on a lot of T cells circulating in the blood of just about everybody. But what there isn't, in our experience, is this T cell subset that is conspicuously double positive for PD-1 and TIM3, and their increase in the CD8 compartment is very, very striking. And as we look at the nanostring, we see other molecules that people have implicated, of course, in T cell exhaustion, including, for example, LAG3. But again, it's selective. I want to point out, it's really important for us to get granular and specific. So, for example, TIGIT, which in other settings is also associated with exhaustion and inhibition, is actually going down in these samples. So there's definitely an exhaustion, chronic stimulation activity, but it's happening coexistent with this extraordinary cell cycling and this extraordinarily striking increase that's severity related in the co-expression of CD38 and HLA-DR, all of which we would say look like acutely activated cells. So there's this is one thing I really want to get across, which is that this disease signature is really very, very dynamic. It's ongoing. It's not chaotic. It's very specific in the traits that are prominent, but it is very dynamic.
Yeah. I want to take you back to the IP10 result.
Yeah.
We were chatting just a little bit before, and that IP10 seems to be a strong signature in viral infection, and we've seen that as well with hepatitis C.
Yeah.
So is it because of the way it's induced, or is it because it's actually required and the immune system is doing a good job here?
Well, it's very hard to know, of course, Nigel. I think one of the problems is, again, that IP10 has been looked at primarily in the context of interferon induction, and it is conceivable that interferon is inducing it here. There is type 1 interferon around, there is type 2 interferon around, and as somebody's pointed out, we haven't yet looked at type 3 interferon, but no doubt there's type 3 interferon around as well. However, what's much more fascinating to me is what is sustaining it. What is keeping this going, potentially in a very selective way. It's not clear, for example, that large numbers of other interferon-stimulated genes that you would see in an ISG signature are sustained in the same way. So I think there are aspects to the pathologic sustaining of IP10, CXCL10, and maybe to some extent IL-6 and IL-10 that we really need to get into. There's a biology or pathobiology of sustained chemokine and cytokine activation that I think we don't understand well, because reasonably enough, all of our understanding of the physiology of cytokines has been in the context of their transient and acute regulation.
Right. You've stimulated a million brilliant questions here. Trying to summarize a few of them. So really, somebody's asking about the T cells and then macrophages and killing virally infected cells. But do you feel, from your perception, that adaptive is reduced and innate is activated? Some people are.
Well, that's. I mean, in terms of adaptive responses, the B cell response, as far as we can see, appears to be fine. I didn't show any neutralizing antibody data, but my colleague Katie Dawes has taken many of our sera and shown good correlation of, high titer spike IgG with virus neutralization. So I think that's going fine. There are clearly peptide-specific, antigen-specific T cells that can be generated. I think it is nonetheless the physiology of mobilizing that adaptive T cell response in a context whereby there is such T cell pathology, whether that's direct or indirect, we cannot say at this point, but I think that's really a struggle. In terms of the innate response, as I say, there are interferons, but whether they're having a fully penetrant effect has yet to be seen. And I mentioned, and we've paid almost no attention to them, which may surprise some people, but there might be antiviral gamma delta and NK cell responses. Those were the two things out of all of the things that were looked at that did actually show some correlation with virus burden measures.
And that leads me on to another question here about the NK cell and the gamma delta. What do you think's really happening there then?
Well, we don't know, but what we do know is that there are many instances, and they're diverse instances, where these where these types of cells get activated in almost an adaptive fashion. For example, in NK cells, as was shown by Louis Lanier and others, Wayne Yokoyama and others by CMV. And CMV likewise has been associated in humans with the expansion of activated V delta 1 cells by groups such as Ben Wilcox's group in Birmingham, Emo Prince in Germany, and Julie Deschenais in Bordeaux. So there are linkages to viruses, linkages to many other viruses than those. Gamma deltas have been linked with West Nile virus, NKs with Hunter virus, etc., etc. So the possibility that these are key contributors to early recognition of virus-infected cells that may actually help asymptomatic individuals clear these things quickly merits a lot of attention. Logistically, those experiments are extremely difficult to set up. As I mentioned at the beginning, one is logistically somewhat limited to looking at individuals where the disease is occurring, which is a good thing, but not necessarily where the very most effective immune response is occurring.
You mentioned about setting them up around the world, and you suggested that these fax panels that really you have now could be useful for other people to do the same.
Yeah, I mean, so we have assumed that whereas not everybody in the world may have a mass cytometry machine that can be deployed at high throughput, they may well have a Fortessa or whatever. I'm hoping BD about to pay for the rest of my reagents for free. So if people have got Fortessas in their labs or their clinics throughout the world or similar sorts of machines, they should be able to run these panels. They're relatively straightforward. Everything is described, and of course, we are available for feedback on the utilization of these. We want people to get as much data globally as possible so that meta-analyses can help us understand what are, as I mentioned earlier, the core components that are seen over and over again, and likewise the value of people highlighting for us where there might, for example, be a cohort that seems to be behaving differently. Is that, for example, because of a variant virus strain that is in that part of the world? So I think we're only going to get to these answers through global deployment of standardized methods.
Yeah. And isn't that what is actually amazing about what's happening? People are working together. And I think it's at the heart of what we're doing here in the IUIS, trying to show people what people are doing so that we can connect better.
It's absolutely spectacular. It's inspirational. In a very perverse way, it's a privilege to be part of it. Of course, we wish that this had never been necessitated, but the one price that we cannot pay is we cannot do all of this and then put it behind us and not be prepared the next time around. So exactly. We have to take the lessons from this global collaboration and recognize that there is a way to be prepared. There are ways in which science can work with sensible public health measures to combat a virus. I'm completely and utterly optimistic that we will do that if people are given very clear instructions as to what it's sensible to do, and if scientists are given the resources and the freedom to collaborate together and to exchange information together openly and constructively. There is no virus that can outwit that combination.
Exactly. And you're right. It would be better if this wasn't happening. Yeah. We could be working away on whatever we were doing before rather than this, but it's the beauty of international collaboration that's hopefully going to help. I just take it back. There's a few questions here about your basophils. That's really fascinating. So many people forget about basophils. Yeah. I'll offend basophil experts by saying that, but they haven't had their day, but somehow or other you're seeing something really interesting with them.
The basophil data are incredibly striking. It's a perfect example of not limiting yourselves too much when you set up these experiments, because of course we might not have thought of including a focus on basophils. So they came out to us. As any clinicians on the line will know, basophil measurements are made routinely in the clinic, but with no offense to anyone, the level of resolution on those is not great. And so, there's a relatively small number of clinical indications in which people look hard at the basophil numbers. What's clear is that flow cytometry here, relatively routinely deployed, can give you resolution on basophil numbers, which is extremely revealing. We don't understand what's going on. In this instance, as I indicated in the talk, it may be tissue homing. Basophils, of course, have been implicated in tissue repair, in lung damage, for example, in asthma and in COPD. But as I mentioned, there's also a body of work on the regulation of coagulation by basophils. And I think one of the things that immunologists need to take to heart is that this is an interesting disease in which probably the highest effect size of comorbidities now relates to vascular disease, to hypertension, and to type 2 diabetes more than it does to, for example, asthma or even COPD. So the prospect exists that an immune dysregulation may indeed be tied in, integrated with vascular pathobiology, and we really don't understand that at all. It is, of course, perfectly possible that these are running in parallel and are not intersecting, but that needs to be investigated. We cannot have a situation where we've got this level of dynamic immune activity going on and that level of vasculopathy and not ask the question as to whether these two are tied in and whether the basophils are giving us a window on that possibility, Nigel, is intriguing.
So we have to wrap up shortly, Adrian, but there's two really interesting questions, and they're kind of in relation to looking after ourselves, I suppose. In relation to smoking, do you think it's protective? And also in relation to vitamin C and vitamin D, do you have any comments on those three things?
I have a very simple comment, which is to keep up your intake of vitamin D, keep up your intake of vitamin C, and don't start or don't stay smoking, even if people tell you it's protective. There's better ways to protect yourself from this disease than smoking.
Yeah, I agree. So, Adrian, is there anything else that you wanted to, maybe we haven't touched on? And I must apologize to people if we haven't got to all their questions, but is there anything else that we haven't really touched on in our discussion that you'd like to mention?
Well, I think we actually have touched upon a lot. I think it's really important for me to say two things: that locally, I'm completely indebted to the selfless way in which postdocs and graduate students and many others just dropped what they were doing and selflessly committed to this project. And as I mentioned, all their names are on our website, and my indebtedness to them is significant. Having said that, that is local. This is a global issue. It's very easy to think that as your first wave sort of washes over your country and then seems to be in decline, it's very easy to ignore the size of the wave in other countries in the world. And we mustn't do that. And I pay tribute to the RUIS for sustaining the global profile of this, because we have to meet this challenge as a global community, not as a set of national communities. It's not a competition. It will be defeated by sensible public health policy and by science and campaigning for the support of science on a global, not a national, not an EU, not a continental level, but on an international level, on a global level, is an obligation that we all have. And I'd like to thank the RUIS for waving that flag, but we have to do even more in support of that.
Adrian, that's perfect to end on. Well, your 13th webinar, I think that's lucky for some. Is that correct?
I don't know. My house number that I live in is number 13. So
oh, perfect. It was meant to be. Yeah. And we've already had 6,000 attendees from all around the world, over 100 countries. So this has really reached out to people. And can I thank you, Adrian, for this extremely informative talk. And it's really going to help. And I know it'll instill collaboration as well in the future.
Thank you, Nigel.
So I just want to mention for people having a look at our website, they can follow our webinars, look at all our webinars that are posted from our previous weeks. And next week, we have Michael Reth from the University of Freiburg, who'll be talking about B cells and coronavirus. And our registration will open shortly. So, Adrian, on behalf of the IUIS and the Frontiers collaboration, I'd like to thank you again. And I'd like to thank everybody for joining us and participating so brilliantly. So goodbye, and of course, take care.
Thank you.